My name is Alice Shiho Huang. I was born in China, but I grew up on the East Coast of the United States for grade school, high school, college. My college was Wellesley College, which is a women's
Oh, I've
college
heard
in
of it.
Boston, Massachusetts. I was a college dropout in order to become an early admissions medical student at Johns Hopkins after 2 years of college. And then I dropped out of medical school in order to enter a PhD program because I became interested in science. I came from a family that had no science background. My father was a bishop of the Anglican Episcopal Church in China. So it was a whole new area for me to explore. And I had no idea that you could become a scientific researcher through medical school or through training graduate school in biology. I got my PhD finally in microbiology, and that's how I spent my career. My first job was at Harvard Medical School as an assistant professor, and I rose to the rank of a full professor in microbiology and molecular genetics. I looked at myself and said, well, now I have tenure at a good university. What do I do next? I really didn't know, but at that time, my husband was interested in a job in New York City, and I thought, well, that could be pretty exciting. And so I did leave Harvard for a job as dean for science at New York University. I of course, joined their biology department, but I learned a lot about administration, went on in higher academics. So I've been in administration ever since, and now I am retired. You never know where your career is going to lead you, actually. I believe that, if you have opportunities, you should just go ahead and take them. And that's really what I did. After being dean at NYU, I joined my husband in sort of a compact that we made between ourselves that I would be helpful to him when he became president of the California Institute of Technology. And so I took on the job as a senior counselor for external affairs. Then, when I finally retired, they allowed me to stay on at California Institute of Technology. We call it Caltech for
Yeah.
short. Help them in taking care of their animal facilities. They needed a professorial type person to head the committee, and also needed someone to help postdoctorals, give them some chance in teaching. And so I got into the pedagogy of teaching and what that meant for people just starting out in science and how to get some experience. And that's been very enjoyable. And I finally even retired from all that.
That's an amazing career. I really hope that one day I can do as much as you did. How did you get interested in molecular biology and genetics?
Well, I really fell into it when I was just starting out in medical school at Johns Hopkins. They wanted each one of us to experience a lab and find out what went on in the laboratories of a faculty member. And I ended up with a biochemist who asked me to look at ATP action, just energy reactions in the muscles of animals. And I started getting into that. I also discovered that I really didn't like that much working with animals and seeing blood, and wasn't particularly interested in very, very sick old people who had a lot of blood bed sores as they stayed in hospitals for a long time, and that I wasn't very good at dealing with situations like that. And I discovered that another professor was working on something called viruses, and that viruses could be very interesting. They are, of course, microscopic and so small that you can't see them with your own eyes. And the laboratories that people worked in, when they worked on viruses, the laboratories were extraordinarily clean and neat.
Yeah.
And I just enjoyed that. And as a graduate student, I made a wonderful discovery. Everything happened. I just followed my nose and asked questions and designed experiments and got answers and published them. It was really, I was just purifying the virus I was working with new techniques that we had all learned. I was using a sucrose gradient, and the purified virus that I had gave me two bands, and I could pull each one of them out separately and look at them in the electron microscope. And one band were beautiful, full bullet-shaped particles. They were cylindrical and rounded on one end and flat on the other, so we called them bullets. And
Oh, okay.
the other band contained what looked like shorter bullets. They were almost round, but if you saw them at different directions, you could actually make a picture of them turning around, showing you the rounded end and the flat end, but they were shorter. And that led me to ask all sorts of obvious questions. Why are they different? Are they both different types of virus particles? Do they have all of the same proteins? And why is one shorter than the other? And it turned out that the shorter ones were deletion mutants, and they had less genetic information, and the genetic information, the amount of it, determined the length of the bullet. I named them defective interfering virus particles because I found that biologically, these little bullets couldn't replicate themselves in cells by themselves, but actually could replicate very well by using some of the information that the larger bullets provided them. And in doing that, they inhibited the growth of the larger bullet. People have gone on to find that there are these defective interfering virus particles in many other viruses and probably in all viruses. And they do play a role in controlling the natural cycle of the virus. It's been
Okay.
hard to actually make a good antidote using these defective virus particles because they're biological agents, and we need to learn a lot more about them and how they work. I predict someday that we will be able to use these defective virus particles and be able to harness them to control the growth of viruses, but going to take a lot of work.
Yeah.
People have worked on it, and we get interesting predator-prey relationships that we can demonstrate in the laboratory in cultured cells. But in the whole body of an animal or in a human, it's much harder to study.
But what kind of traits are you looking for in a student? What makes someone stand out to a professor?
It's very hard. I have admitted very few individual undergraduate students to work in my laboratory. I've found that one of them, I said, have you had any experience in the research? Did you do research in high school or started when you were a freshman in college? He said, everyone asks me that. And how can I get started in research if I haven't had any research
Yeah,
experience?
yeah.
And so finally I asked him, I said, well, can you cook a meal? And he said, yeah. He says, my mother taught me to do some cooking. And I said, do you enjoy it? And he said, well, I did it. So I said, well, if you've cooked and you know something about measurements and so forth, you would do well in a research lab. But I had no way to measure how smart he was, except that he was a Harvard undergraduate. So at least use that. So I look for people who are basically smart, who use common sense, and who like experimental methodologies. And we do most of our experiments in molecular biology with biochemical methods, and now more and more actually with mathematical methods. And I will sometimes ask a student what their grades were senior year in high school in math and in English. If I lived in China, I would ask them how good their grades were in math and Chinese, because those are the two traits that is a prerequisite for people to become successful. You have to have a certain amount of competitiveness. I call that the killer instinct. And I've had some students who are extraordinarily smart, but they lack a real killer instinct. And I wish I could have taught them that, but that's not something that is hard to teach. It is something that's hard to teach anybody.
Yeah.
They have to really want something and want to get there first, or be the best that you can.
Where do you see your field going into the future? Like, what do you think students can focus their attention on?
Biology goes through cycles, and when I entered, it was at a time in which the very basic building blocks of biology weren't known, and they were just being discovered. It was the time of discovering the structure of DNA, the ability to use radioisotopes as tracers, a whole bunch of new methodology to look at things in a molecular biochemical way. So we followed the direction of simplifying things, and viruses at that time were considered a simple biological structure. It had a few genes in a package, and you can manipulate it. Where do I think virology is going now? I think it's going into an area. If I were to start now, I would want, well, I have a good background in virology, but I think that the interaction of the virus and the host is probably going to be the most challenging in the future. And you would want to choose systems that are important. So, what part of the host would be most important? And what is the most exciting thing that's going on in human biology now? That's really in the brain, in the nervous system. Neuroscience, coupled with viruses as a probe, can be very exciting. That's the area that I would pursue now if I had just stepped into it and had the virus background.
Thank you so much for your time, and I really enjoyed talking to you.